Good evening, everyone. I welcome you all to the third live session of Bioinformatics Algorithms and Application course. So, this will be a combined live session for week 3, week 4 as well. Before moving on to the session, how many of you have watched the week 4 videos which are out? Can answer by through chat box or raising your hand.
Sir, can you repeat the question? I didn't get it.
How many of you have watched the week 4 videos, the NPTEL lecture videos?
Sir, I am personally left with 2 of them.
Okay. Okay, I'll start with the lecture. So, about the session, today's session, I won't be using the Google Meet polls. Try to answer the questions using the chat. And coming to the session, how the session progress. Initially, I'll be discussing about summary of week 3 lectures and week 4 followed by week 4 lectures, and we'll try to solve some of the sample problems based on the week 3 and week 4 content. Then I'll be spending time for the questions. Okay, starting with the lecture 1. So previous week and before lectures we have discussed to measure sequence similarity as well as sequence alignments. So as we discussed, it is possible to measure sequence similarity in many different ways, such as counting the number of differences between them, like measuring the Hamming distance just to differ how sequences are different, and counting the number of insertions or insertion, deletions and substitutions required to make two sequences identical. Or we can also calculate the percent identity or just use an arbitrary scoring system for matches, mismatches or indels, nothing but insertions and deletions. So all these methods yield a measure of relationship between sequences, but none reflect any biological association between them. So in bioinformatics, we are interested in an evolutionary relationship of DNA and protein sequences, except in case of sequence assembly, where measuring sequence errors and separating repeats are central. Coming to the biological sequences of DNA or protein, these sequences may be more or less similar by sheer random chance, and consequently need a method to distinguish a random similarity caused by the evolutionary relationship. Just give me a second. Yeah. So these biological sequences may be more or less similar by chance, and consequently we need to have a method such a way so we can distinguish a random similarity from similarity caused by the evolutionary relationship. In other words, we wish to know whether sequences are homologous, that is, have a common ancestor, and in particular, whether sequences have same function despite not having identical sequences. Being able to determine if two sequences have the same function is useful in appraising the function of an unknown protein and gene by comparison to a known one. The amino acid sequence of a protein is crucial in determining its structure and function, and many amino acid mutations resulting in the change amino acid having similar physiological properties may not alter the protein structure in any functional critical way. Further, by enumerating the frequencies of these changes, we can construct a scoring systems, like see based on the mutations that are accepted by nature or that come back on the natural selection. So we can observe some frequencies based on the scoring systems. So these scoring systems are used to determine the relative score made by matching two characters in a sequence alignment. These are usually, so these relative frequencies or scores, what we call, are usually represented as a logarithmic of the likelihood of two characters being derived from a common ancestral character. So there are many types of scoring matrices for amino acid sequences, nucleotide sequences and codon sequences, and each is derived from an alignment of known homologous sequences only. So these alignments are then used to determine the likelihood of one character being at the same position in the sequence as the another character. So this gives the probability values of being observing a mutation in between these. So in generating a scoring matrices, first research was done by Margaret Dayhoff in 1970s and colleagues, and later by Henkoff in early 1990s, which resulted in PAM and Blossom substitution matrices, and they are most widely used today. So in this lecture, we will be discussing their construction and how to use those. So let's start with the PAM. As the name describes, PAM stands for Point Accepted Mutation Matrix. So these were initially introduced by Margaret Dayhoff in 1978, and the calculation of these matrices were based on 1572 mutations in the 71 family. So in 71 closely related proteins. So these proteins are studied have higher similarity with their next generation. So these protein alignments were included to only when they display at least 85% identity. So why these alignments, or why the participants in the alignments have higher identity? Because having a higher identity is reasonable to assume that any aligned mismatches were the result of single mutation event rather than several at the same place. So this selecting the higher higher identity homologous sequences will, so we assume that these are the effects of the single mutation event. So PAM mutation matrix is a matrix used in bioinformatics to quantify the evolutionary distance between two protein sequences. It is used in the sequence alignment algorithms to score the likelihood of different possible alignments between two sequences. These matrices are constructed using a statistical analysis of large number of closely related protein sequences. The underlying assumption is that mutation occur at a relatively constant rate over time, and frequency of amino acid substitutions can be used to estimate the evolutionary distance between sequences. Generally, the PAM matrix is a 20 by 20, the 20 cross 20 matrix, where each cell represents the score assigned to a substitution of one amino acid for another. So generally, we observe half matrices because this 20 by 20 matrix has an symmetry. So most of in situations we represent them as a half matrix. So the values in the matrix are represented in the term of a PAM units, which represent the estimated number of mutations per 100 amino acids that have occurred during the evolutionary history of two sequences. For example, when we see a score of 1 in the PAM matrix, that means that the substitution is expected to occur once in every 100 amino acids. Similarly, when we see a score of 5 means the substitution is expected to occur once in every 20 amino acids. So the PAM matrix, as I mentioned, is widely used in bioinformatics for sequence alignment and homology searching. So let's understand how this PAM matrix is constructed. So the construction of a PAM matrix involves 3 steps. That are, first step is to create a matrix of observed amino acid substitutions, that in an alignment we see what are the possible substitutions that were observed, and converting these observed matrix into the matrix of log scores, and finally normalize the matrix to the PAM1 matrix, which represents the expected number of substitutions per position after 1 unit of evolutionary distance. So, okay, discussing them deeply. So, the first step is create a matrix of observed amino acid substitutions. The first step in creating a PAM matrix is to gather a large number of homologous protein sequences and identify the amino acid substitutions that have occurred in their evolution. So, if you observe here in the given sequence alignment, we have seen the amino acid substitutions in the third position and the fourth position where leucine has been substituted. So the first step is observing and making a matrix of these all possible substitutions. So the resultant matrix has been given here. So I have given a short matrix of only 5 amino acids. Generally the matrix will be of 20 by 20 amino acids. So this resultant matrix will contain the observed frequency of each amino acid substitution. For example, the matrix here shows the number of times each amino acid has been substituted for another amino acid. For example, the frequency of substituting alanine and cysteine is 107, where you can see the second column first element. So these correspond to the number of times alanine has been substituted by cysteine. Then this matrix has been converted into the matrix of log odd scores. So how this is converted is, this is converted by using a formula where S of ij corresponds to the amino acid 1 and amino acid type 2, and where O is the observed frequency of substitution of amino acid I with the amino acid J, and EI is the expected frequency of amino acid I, and EJ is the expected frequency of amino acid J. The expected frequency of an amino acid in a protein can be calculated using the protein length multiplied by amino acid frequency. Here protein length means the total number of amino acids in the protein, and amino acid frequency is the frequency of specific amino acid in the reference of dataset being used. So this can be calculated by dividing where amino acid frequency can be calculated by dividing the number of occurrences of amino acid in the reference dataset by the total number of amino acid in the dataset. The next step is to normalize the matrix to the PAM1. So what type of scores? The matrix here scores positive score, a 0 score, as well as a negative score. So what these scores represent? Positive scores in the matrix denotes substitutions occurring more frequently than would have occurred by a random chance or expected among evolutionary conserved replacements in the data set of homologous sequences. A 0 score in PAM matrix means that frequency of amino acid substitutions found in homologous sequence dataset is equal to that of expected by chance. In this case, the relationship between amino acids is weakly similar at best in terms of physiochemical properties. A negative score corresponds to the substitutions found in homologous sequence dataset less frequent than would have been. Yeah, I have seen a hand raise. Do you have something to ask? Yes, you can ask through the. Since I see many people are unable to, they're not turning off the mic, which causes disturbance in the recorded lecture, I turned off. So you can ask your question using the chat box. Generally, we'll be discussing here summary of the important lectures or important concepts that will help you to solve the sample problems that we'll be solving later. So we'll be discussing those here, those topics in a brief way. And even those, apart from those actual lecture videos, our lecture videos will also be available in the NPTEL website under the problem solving session. Okay, moving forward. Here the negative score corresponds to the substitutions found in homologous sequence of dataset less frequent than would have occurred by random chance. Okay, here let me move on. So here is an task, given alignment here and given the matrix is a PAM 250 matrix. So can you try to score this alignment and mention the answer in the chat box? So I have given alignment here where the sequence 1 is TAHGK and sequence 2 is YSDJD, and I have given a matrix also here. So using the values given in the matrix, can you score this alignment? So I'll wait for a minute. So given alignment here and matrix here, can you people score this alignment? No answers. 4. Okay, I'll move forward. 4. Okay, I'm getting some answers. Yeah, the first step is we need to identify the matrix score for the observed combinations. So here the combinations will be T and Y. So the score of T and Y in the given PAM matrix is -3. So where is P is the fourth element, and the score for A and S is 1, and H and D is 1, and score for G and G is 5, and finally the score for K and D is 0. And finally sum up all these scores to get the score of an alignment, which is 4. So these have been taken based on substitutions that are observed in the given alignment, and what is this next step is to find out the score for the alignment, then sum up to get the final score. Okay, moving forward to the Blossom matrix. These are very popular scoring systems used in bioinformatics, and they are substitution matrices that describe the likelihood of amino acid substitutions between different protein sequences. So these Blossom matrices are constructed by dividing the sequence into blocks of similar sequences. Even the Blossom expansion stands for block substitution matrix, and once these blocks of similar sequences have been constructed, then they go for calculating the frequency of substitutions between the amino acids in each block. This is similar to that. This is also a two-dimensional array where each row and column has different amino acids. The matrix element represents the scores assigned to each substitution event, with higher scores indicating more likely substitutions. Here in Blossom matrix, the positive score indicates a favorable substitution and a negative score indicates an unfavorable substitution. So the Blossom matrix is typically used in sequence alignment algorithms such as BLAST, which is nothing but basic local alignment search tool, to compare protein sequences and identify the regions of similarity. So briefly, Blossom is a matrix that reflects probability of amino acid substitutions in related proteins based on the frequency of substitutions observed in the set of aligned protein sequences, and is created first by constructing a multiple sequence alignment of a set of related protein sequences, and from this alignment a frequency table is generated that indicates the number of times each pair of amino acid appears in the aligned sequences. So this frequency table is then converted into a scoring matrix that assigns a score to each possible pair of amino acids based on their frequency of occurrence in the alignment. The major steps in the construction of an Blossom matrix include identification of protein families. So this Blossom matrix is constructed using a set of protein families that share a certain degree of similarity. The most commonly used set in the blocks is the blocks database of protein families. The second thing is alignment of the protein sequences. The protein sequences in each family are aligned and frequency of each amino acid substitution is calculated. Finally, converting these frequencies to scores. The substitution scores are calculated based on the observed frequency of each substitution. This score reflects the likelihood of one amino acid substituting other, and even these are also log odd units only. Here the given formula where f of ij is the frequency of substitution of amino acid I from amino acid j, and fi is the frequency of amino acid I, and fj is the frequency of amino acid j, and n is the total number of amino acids considered. Okay, let us move about the dynamic programming methods. So we have discussed these dynamic programming methods also, that is global alignment and local alignment. So global alignment, which attempts to align every residue in the sequence and are most useful when sequence in the query are similar and are of roughly equal size. A general global alignment technique is that Needleman and Wunsch algorithm, which is based on dynamic programming. Coming to local alignment, they are mostly useful for dissimilar sequences that are suspected to contain regions of similarity or similar sequence motifs within their large sequence context. So the Smith-Waterman algorithm is the general local alignment method based on the same dynamic programming scheme, but with additional choices to start and end data place. So we will be studying these alignments later. And there is one more, a hybrid method known as semi-global or global, the short form for global and local. So which search for the best possible partial alignment of two sequences. So this can be especially useful when the downstream part of one sequence overlaps with the upstream part of the other sequence. In this case, neither global nor local alignment is entirely appropriate. So a global alignment would attempt to force the alignment to extend beyond the region of overlap, while a local alignment might not fully cover the region of overlap. Okay, let's start with the Needleman and Wunsch algorithm. So this algorithm is used to align the protein or nucleotide sequences, and it was one of the first applications of dynamic programming to compare biological sequences. This algorithm was developed by Needleman and Christian D. Wunsch and published in 1970. So this algorithm essentially divides a large problem into series of smaller problems, and it uses solutions to the smaller problems to find the optimal solution to the larger problem. It is also sometimes referred to as optimal matching algorithm or the global alignment algorithm. So this algorithm constructs a matrix to store the alignment scores, and it uses it to find the optimal alignment. So here are the steps that are required to construct a Needleman-Wunsch matrix. The first step is initialization. So in the first step is to create an n plus 1 cross m plus 1 matrix. So where n and m are the sizes of the sequences or length of the sequences aligned. The first row and column of the matrix are filled with gap penalties. That is, in the first row and first column, we are aligning our matrix with a gap, which represents the cost of opening a gap in the alignment. And the topmost left corner of the matrix is set to 0. That is, you can see here f of 0, 0 in the matrix. So where the first 0 represents the row and the second element represents the column. The next thing is fill the matrix. Starting from the second row and column, each cell of the matrix is filled by row-wise or column-wise manner. And the score in each cell is calculated by considering 3 possible ways to get into a cell. That is diagonal, left and up. The diagonal score is the score of aligning 2 characters in the sequence. The left and up scores are the gap penalties. That is the penalties for introducing the gap. So when we consider a matrix or a cell in a matrix, so the value of that matrix is calculated in 3 ways. That is from up cell, the cell which is above it, a cell which towards its left, and towards the diagonal. And finally is the traceback. So once we fill this complete matrix, the optimal alignment can be found by tracing back through the matrix from bottom right corner to the top left corner. Okay, now we'll see a practice problem. So as I mentioned, the top left is always initiated with these 0, so that now we are aligning to this gap penalties, and followed by that we are filling the first column as well as the first row with the gap penalties. So as I mentioned, when we move towards up or left, we do with the gap penalties. So 0 plus gap penalty gives the value under the cell A. Minus 2 plus gap penalty gives the value in the cell under the cell G. Likewise, we initialize the matrix, then we fill the matrix using the Needleman and Wunsch rule. So they have called it as update rule, where update rule for a cell mentions that the value of a cell corresponds to the maximum value obtained by the previous 3 cells. Finally is the traceback. So since this is a global alignment which considers entire sequence as alignment, we try to take from the last cell and move towards the first cell in the matrix. Okay, now we try this an alignment. Yeah, okay, how to do the traceback. So once you see, when you consider this cell, I hope my pointer is visible. Yeah, so these arrow marks. So when we have to calculate a value for a cell, it has 3 ways to come. And this arrow mark here in the last matrix corresponds to the, so this value of -1 has been taken from the calculation of 0. Yes, since this is a global alignment, even if you get a maximum value in the between the cell anywhere, also we need to do it from the one corner towards the other. And regarding the traceback, given this rule where the value of a cell is calculated from the maximum of the previous 3 cells, that is adjacent 3 cells, so traceback will be towards that. So here when you see the last -1, so this value of -1 has been derived from the calculation done on the 0 cell, which is in blue color, highlighted blue color. And when 0 calculation, the value of 0 has been obtained from the -1, and this value of -1 has been obtained from the 1, and this value of 1 has been obtained from 0. So traceback is done in such a way that the corresponding cell value has been obtained from the calculations performed on which cell. So we point this arrow towards that cell. So when these arrow marks are moving diagonally, that means it's a match or a mismatch, or otherwise if they move vertically or horizontally, means it is a gap, introduction of a gap. So now let's try with an, I hope I'm clear, or you want me to repeat again? Okay, thanks for the response. Okay, let's try for this alignment of sequence A and B. I'll wait for some time for you guys to note down the sequences. Okay, let's move forward and I'll do the initialization. Yeah, okay, I'll be explaining using this example now, how the traceback is done. I'll do that. So now I have created a matrix here. I put one sequence on the top and other sequence on the left. And I first topmost left cell, I started with 0 and 0. And rest all cells which are aligned to the gap, I have added them to the gap penalty. So no doubt till here, right? Everybody is clear how I got this initial matrix. Yeah, once this is done, I have filled next some of these rows have been calculated, and I want you guys to calculate what is the value for the cell S42. That is the question mark where you can see. So can you guys calculate that? Calculate and let me know the value through chat, and we'll solve for this cell. I hope now the screen is visible. Okay, let us move on to the solution. Yeah, so the given neighbors for the cell are 0, -11 and -14, and we have asked to calculate what is the value of this cell. So this value of the cell can be calculated from three ways. That is, from 0 cell we can calculate, diagonally from -11 we can calculate, and from 14 we can calculate. So let's label these cells as 1, 2, 3. So in the case of 1 and 2, we use gap penalty. In case of 3, we use match or mismatch score. So considering the gap penalty, the value in the cell 1 is 0 plus gap penalty. So which equals to 0 minus 3 is minus 3. Coming to the cell 2, the value in the cell 2 is minus 14 plus gap penalty. So which gives minus 14 minus 3 is minus 17. Third case is, before moving on to the third diagonal case, we need to see whether these are match or a mismatch. So the corresponding top sequence element is A and the leftmost sequence is T. That is, this is a mismatch. So what we do here is a cell value plus mismatch score, which equals to minus 11 plus of minus 5, which equals to minus 16. Here we need to get maximum of minus 3, minus 17 and minus 16. So here the answer is minus 3. So the answer is minus 3 and it has come from the topmost 0 cell. Okay, similarly, can you guys calculate for S5, 5 cell? Yeah, I want you people to calculate value for the fast answers. Okay, I've seen some answers. Okay, I'll move on to the solution. Okay, we'll try to solve similarly. So first we'll try to identify what are the neighboring cells. So for our calculation cell, the neighboring cell is 16, 19 and 16. And this value is calculated like this, value is calculated like this, and this is diagonal. And let us again label them as 1, 2, 3. So coming to the cell 1, we take what is the value of cell plus gap penalty, which equals to 16 minus 3 equals to 13. Similarly, in case of second, we have 16 as cell value plus gap penalty, which gives rise to 16 minus 3 is again 13. Next, in third case, so before moving on to the third case, we should see whether it's a match or a mismatch. So the condition here shows is a match. So what is the value in the cell is 19 plus match score 18, that equals to 27, and maximum of these two values is what we had here, it is 27. Sorry for the alignment error here. So actually it's a mismatch condition where we have to do minus 5, because the top sequence is not aligned properly. So this is what the final matrix we get here, and has yes please. No, the answer won't be 13 because yeah, it's always a gap. Gap penalty is what? See, when we are moving from horizontal cell or the vertical cell, we always consider gap penalty only. We won't take other match or mismatch. Match or mismatch only comes in this third case. In 1 and 2, it's always gap penalty only. This is constant. Okay, once we have this final alignment, someone have asked me how this traceback has come. When you consider this last cell, which is 27, sorry, 27, from 27 we are moving diagonally towards 32. So that means here the chances of mismatch or match is high. So we add A and C, and from 32 cell from 32 cell, this value of 32 has been derived from 24. Again we are moving diagonally, so we add G and G as usual. So there is no, unless we are moving horizontally, there is no need to have a concept of gap. From 24 to 16 also we are moving diagonally, and in the case of 16, the value of 16 has been derived from the cell 19. That means we are moving horizontally, so towards 19. So there is a, it says that there should be a gap indicated in the place of T here. So you see, once this alignment is done, so this 19 corresponds to the C here, and here it is aligned to a gap. So here its movement is like this. So here we introduce a gap. In all the cases the movement is along the diagonal, but when we are moving here, this indicates the place where a gap has to be introduced. So this corresponding C and gap. And we can also cross verify. Yes, I'll be discussing week 3 and week 4 today. Week 4 is just phylogenetic today. Yeah, and coming to the alignment score. So the last value in this matrix is called as an optimal alignment score, and this value can also be confirmed by calculating from the sequence. So from the sequence we see this is the alignment that we have obtained, and in the alignment you see C and G is a mismatch, so you add -5, and A is a match score, so you add match score, and when you introduce a gap, add a gaps penalty. Like how you calculate this for the alignment, value is 27 again, and the value that we see at the end of the matrix is also 27. Okay, moving forward to this with Smith-Waterman algorithm. So this performs the local sequence alignment, that is for determining the similar regions between two strings of nucleic acids or proteins. Instead of looking entire sequence, the Smith-Waterman algorithm compares segments of all possible lengths and optimizes the similarity measure. As such, it has desirable property that it is guaranteed to find optimal local alignment with respect to the scoring system being used. The main difference between the Needleman and the Smith algorithm is the negative scoring matrices are set to 0 here. So in the Smith-Waterman algorithm, there is no negative values are allowed. If we see any cell correspondingly having a negative value, we make it 0. And here in this algorithm, the traceback process starts from this cell having the highest value, because it looks for only the local similarity, not the entire sequence similarity. So this algorithm has several steps similar to that Smith algorithm, where we initialize the scoring matrix, then followed by scoring and traceback. As I mentioned here, the traceback here starts from the highest score and it continues recursively until it is encounters with 0. Yeah, let's do this practical for this. So as I mentioned here, if you observe the global alignment thing, the gap penalties has been negatives, but here we have extra case. 4 conditions have been given in Smith-Waterman algorithm. So if any of the cell value is less than 0, so we take 0 as the value. So that's why, since gap penalty is -3, so all the first row and the first column has been to zeros. So can you people tell me what is the value in the question mark? Yes, the value is 7. So can anyone tell from where the alignment traceback will start here? I told in Smith-Waterman, the traceback will start from the cell having the highest value. So what is that cell? Yes, it starts from 37 and this recursively goes on until it encounters 0. If it is global alignment, we take from 32 to this 0. Since this local alignment is only worried about the local similarities or the small subset similar regions, so we take from the cell having maximum values until it encounters the 0. Similarly, this highest cell value corresponds to the optimal alignment score as well, where you can see 37 here when we calculate the alignment and 37 here. Coming to the other two techniques, which are FASTA and BLAST. So BLAST and FASTA are two similarity searching programs that identify homologous DNA sequences and proteins based on the excess sequence similarity. So we have different variations in BLAST program. So BLAST, which uses heuristic programs to align the query sequences with all the given data sequence in the database. So the objective of the BLAST is to find high scoring uncapped segments among the related sequences. And there are different variants of BLAST, such as BLAST-N, which compares a nucleotide sequence with the nucleotide sequences, and BLAST-P, which compares protein sequences with the protein sequences, and BLAST-X, which compares nucleotide sequences against the protein sequences, TBLAST-N, which compares protein sequences against the 6-frame translations of the nucleotide sequences, tBLASTX, which compares the 6-frame translations of nucleotide sequences against the protein sequences. And next method is the FASTA. So FASTA stands for Fast All. So it was the first database similarity searching tool developed, followed by the development of BLAST. So FASTA uses a hashing strategy to find the matches for a short stretch of identical residues within a length of k. So the string of residues is known as k-tuples or k-tubs, which are equivalent to the words in BLAST, but are normally shorter than the words. Next thing is multiple sequence alignment, which is a method for aligning multiple sequences. Till now we have discussed aligning a 2 sequence at a time, that is dot plot, Needleman-Wunsch algorithm, or Smith-Waterman algorithm. We have seen 2 sequences at a time are only considered. So multiple sequence alignment is a step forward to it. So in this kind of multiple sequence alignment, it first aligns the most closely related pair of sequences, then goes for the next most similar one, and so on, as it contains more complicated and sophisticated algorithms. So these multiple sequence alignments are applicable for detection of variable regions between the sequences that include phylogenetic analysis, or for detection of homology genes between new sequences. Muscle, ClusterW, MAFT, and tCoffee are some examples of multiple sequence alignment tools that are frequently used. Okay, now we'll try to solve some problems. So here is the first question. The first question is, gaps are added to the alignment because here is the first question. You can answer it using the chat box. So the gaps are added into the alignment. So what is the purpose we are using gaps in an alignment? How they are useful? Okay, the answer is here. Okay, yeah, gaps play an important role in sequence alignment, as they allow for the proper alignment of sequences that contain insertions, deletions, and other types of mutations. Here are some key reasons why gaps are important in sequence alignment. They allow for better alignment accuracy. That is, gaps can help to accurately. Yeah, I see that. The gaps can help accurately to align 2 sequences that have different lengths or that contain regions that do not match perfectly. So how they help to align the sequences that have different length? By inserting gaps in one or both sequences. Next thing is reflect evolutionary relationships. Gaps can also help to identify evolutionary relationship between sequences, insertions, and deletions. For example, are often used to infer evolutionary events such as gene duplications, divergence, and gaps in sequence alignments. And gaps can also facilitate for functional prediction and also help in experimental design. Okay, before moving forward, let me turn on Q&A for any questions. So I have turned on Q&A session. If you have any questions, post it there. Then we'll move on to the next session. Yeah, the next question is regarding the PAM matrices. So the question is the PAM matrices are derived from evolutionary model, whereas the Blossom matrices consist of entirely direct observation. This is an true or false question. No, don't worry about the numbering because this is the common set of questions that are prepared for the week 3 and week 4. If you see a number missing here, the number will be appearing in the week 4 question. So now I wanted to discuss week 3 questions and then I'll move forward to the week 3 explanations and solving. So now we are solving question 3 here. So that our PAM matrices are derived from evolutionary model, whereas Blossom have been from entirely direct observation. So here the answer is true because we all know these are the PAM matrices are based on the idea that occur at relatively constant rate over the evolutionary time. So we build an evolutionary model by aligning and then we try to get the relative constant rate of substitutions that we see. Coming to the Blossom, these are constructed from a larger or diverse set of protein sequences and are designed to score alignments of more distantly related sequences. That is why these are the direct observations and PAM matrices are from evolutionary model. So the next question is a fill in the blank kind of question. So dynamic program involves, so you have to mention what is could be this first word, second word, third word and the fourth one. So these are the steps in dynamic programming that we discussed. You have to fill all the 4 answers here. So what will be the first one that is related to dynamic programming? What is the second one that is related to dynamic programming? And third one that is related to dynamic programming, and fourth. Yes, the first one corresponds to the initializing the matrix by filling with the by filling the top left cell with the 0, followed by the filling first row and first column using the gap penalties. Second one is matrix filling. So the Needleman algorithm or the Smith-Waterman algorithm has their own way of filling their matrices. Next is the traceback. Once matrix filling is done, we start with the traceback, and finally we get an optimal alignment. Next is a fill in the blank question. So in which alignment 2 sequences to be aligned are assumed to be generally similar over their length, and in which alignment does not assume that the 2 sequences in question have similarity over the entire length? Yes, global and local. Yeah, it's global and local alignment. Right answers. Thanks for answering. So next question is, which one of the following does not describe local alignment algorithm? the answer is right. So if I change the similar question, so which one of the following does not describe the global alignment algorithm? Can you guys answer? So now I'm changing the similar question. Which one of the following does not describe? It could be multiple answer as well. So what could be the one that doesn't describe, does not describe? Yes, the answers would be B, C, D, because we never set a negative score to 0 in global alignment, and also first row, first column are never set to zeros because we use the gap penalty over there, and the traceback in the global alignment will begin with the one corner to the other corner. So next is a question of a database. So when we have a database such as a BLAST or with search to them, so the quality and the time of a database searching depends on the answer is both A and B, because the number of sequence in the database increases the complexity of the search algorithm, and size of the query increases the complexity of the time in terms of time. Okay, given 5 different basic BLAST programs, so answer these questions. So first answer for the first one, so we'll move one after the other. So I have provided the table at the below for your reference, so you can kindly refer to that table and answer the questions. Okay, here the first one is, you have protein sequences and you would like to know what are the other similar proteins to it. So where our query sequence is protein and database search is also protein, so it comes BLAST-P or protein BLAST, both are same. So next thing is, you have a DNA sequences and you will need to search for the other DNA sequences that encodes the same or similar protein. So here is the BLASTX. So third question is similar, like we have a DNA and we need to search against the DNA sequences. So its simple question is nucleotide BLAST or BLASTM. Finally, we have a protein sequence and we wish to search against a DNA database to find the genes. That will be dBLASTM. And next is again a fill in the blank question. The question is, BLAST uses dash kind of a matrix or method to find a matching words, whereas FASTA identifies identical matching words using what method? Has most of the biology people or bioinformatics people, they use BLAST and FASTA. They should know what is the background method they use for. Yes, the answer is correct. The BLAST uses substitution matrix, and whereas FASTA uses hashing procedure. Okay, from here on I'll be moving towards the phylogenetic analysis. So coming to the phylogenetic analysis, this is part of the week 4 lectures. The study of the evolutionary development of a species or a group of organism or a particular characteristics of an organism, or to analyze the relationship within or among the species, is known as a phylogenetic analysis. Generally, phylogenetic analysis has the branching diagrams that are made to represent the evolutionary history or relationship between the same or different species. The diagram that is used during the phylogenetic analysis is known as a phylogenetic tree as well. And this phylogenetic analysis is used for different purposes that include a collection of biological diverse information and information of genetic classification, as well as learning on different developmental stages that occur during evolution. So when we take a phylogenetic tree, it is compared basis on the leaves or tips, nodes and branches that were nearby nodes. So in the phylogenetic tree, species, population, individuals or trees are represented by the leaves, and these leaves are connected to nodes with the help of a branch. The flow of genetic information between subsequent generations is determined by the branch and genetic change. So the phylogenetic trees can be rooted and or unrooted, as well as scaled or unscaled, depending on our study requirements and what kind of tree we require. So rooting of the phylogenetic tree is essentially required for better understanding of the directionality of the evolution. So let's try about different phylogenetic trees. So there are different types of phylogenetic trees, which include rooted phylogenetic tree and unrooted phylogenetic tree. A rooted phylogenetic tree is a useful diagram that represents evolutionary history. This tree has a basal node that represents the common ancestor of all the groups of the tree, and this basal node is called as a tree. Here you observe Z has a root. And this node is also considered as an oldest point in the tree and represents the last common ancestor of all the groups included in the tree. Hence, the direction of evolutionary time is shown by a rooted tree. In order to find common ancestor or species ancestors, tracking back the basal node is necessary from single species of a rooted tree. Coming to the unrooted tree, so this tree lacks common ancestor as a basal node are known as an ancestor. So you can't see any common node or where a kind of evolutionary path has started. So these kinds of tree doesn't show the origin of evolution of the groups, but they show the relation between the organisms without involvement of direction of evolutionary timeline. Next, let's study about different parts of a phylogenetic tree. Coming to the branches, so these are the paths of the genetic information transfer from one generation to the other, and genetic changes are noted by the branch length. Coming to the nodes, these are at the end of the branches, and there is a node that represents the sequence at various points in evolutionary history. Next, tips are also known as the external nodes, representing the sample sequences or interesting species during the construction of phylogenetic tree. Next is the internal nodes. These are the junction points of the branches, or the point where more than one branch meets, and represents that the ancestral sequences. Next, we study about the different methods that are available for the tree construction. So first one are based on the distance-based methods, and second type is the character-based methods. And coming to the distance-based methods, they rely on the measure of genetic distance between the sequences that has been classified. Here, a distance is the fraction of mismatch between the two aligned position of sequences, where the gaps are either ignored or counted as a mismatch. The distance method describes the distance between each pair of the sequence, and from sequence query it attempts to construct an all-to-all matrix. Thereby, the phylogenetic tree constructed from this method places the closely related sequences under the same interior nodes, and their branch length produces the distance between each sequence pair. The widely known distance-based method is a neighbor joining method. This method is fast and can be used with a variety of models as compared to that of character-based methods. And this neighbor joining method provides an unrooted and single resultant tree. Next is the character-based methods. The two widely known character-based methods are maximum parsimony and maximum likelihood methods. Maximum parsimony is one of the methods for identifying the potential phylogenetic tree in which the observed sequence data are explained with the help of smallest total number of evolutionary events. So when every possible type is not equally likely, then this approach is very useful. For example, when amino acid nucleotides are known to be more mutable, simple enumeration is the most native way of identifying the parsimonious tree. That is, considering the succession of each possible tree and scanning for the tree with the smallest score. Next is the maximum likelihood method. The maximum likelihood method is use of standard statistical technique for inferring the probability distribution to assign the probabilities to a particular possible phylogenetic tree, and requires a substitution model to access the probability of particular mutations. So this kind of method, such as maximum likelihood, is well suited for the analysis of distant related sequences, but they are computationally expensive for performing for more sequences. Next, let me start with a quiz. So this is kind of a match the following. So consider this as 1, 2, 3, 4. There's A, B, C, and D. So these are the different terminologies that we discussed just before from a phylogenetic tree. So what these terminologies represent actually? Okay, let me move towards the answers. So the root represents the origin of evolution, leaves represents the current organisms, the branches represents the path of evolution or relationship between the organisms, the branch length gives the evolutionary type. All the answers are given are correct. So given a Newick notation, so here we have given a tree. So can you represent them in a Newick notation? Unique notation is a kind of a text format rotation, and this concept is discussed by sir in the videos. Yes. I have got the answer, but the answer seems to be incomplete. Yeah, yes. Now the answer is correct for the A. Yeah, where we try to keep similar nodes together. Here, so what we have the branch lengths. So when we have the branch lengths, we represent them in this scan. So if the tree is scale, the branch lengths are indicated immediately after the taxon name. You can see without branch lengths the A, and with branch lengths it's B. So moving on to the next question, how many rooted and unrooted trees can be obtained using 4 sequences? I have given formula here. If in case someone doesn't remember the formula. Yeah, the option A is correct. Like I think it should be 15.3. Yeah. So this is the formula, and for given n value, these are the possible number of rooted trees and unrooted trees that can get. Next is regarding the UPGMA method. So which of the following is untrue about the UPGMA method? So there are some main steps in the UPGMA method which calculate the pairwise distance between all the sequences in the database, and this can be done by using any of the standard distance metrics such as Hamming distance or Jukes-Cantor distance. Then they create a matrix to store the pairwise distances between all the sequences, and then we start clustering two sequences at a time with the smallest distance to each other into a single group. And calculate the distance between this group and the other remaining sequences using arithmetic mean of the pairwise distances. So these are repeated until all the sequences have been clustered into a single group. So they use clustering information to construct a phylogenetic tree where the length of each branch is proportional to the evolutionary distance between two or more groups. Yeah, answer is B. So here is a simple question. A bifurcating branch point in a phylogenetic tree is known as. And I forgot to mention before, one of the main advantage of UPGMA method is its computational efficiency, as it can handle large data sets with reasonable amount of computational resources. However, it has some limitations such as the method is sensitive to outliers and assumes a constant evolutionary rate over time, which can result in inaccurate phylogenetic trees if assumptions are violated. Okay, here is the fifth question. Yes, it's no. Yeah, you can take this as a task. Given a amino acid sequence, can you compute the amino acid occurrence, amino acid composition, and amino acid residue pair preference? So here I have given the amino acid occurrence and composition. If you see in the given sequences, there are 4 alanines, 3 arginines, and so on. So take this as a task. Here is the next question. Calculate the molecular weight of the given peptide. And in the brackets I have mentioned the molecular weights of each amino acid. Yeah, these are just basics question, because next session will be again on next Monday. By the time your assignment will be also out. So these are some basics questions we'll be trying to take here in this session. Yeah, I'll explain this. Yeah, given the sequence here, for the amino acid occurrence, we need to calculate for the occurrence of 20 amino acids. So when we count for alanines, we can see 2 alanines here, and we can see 2 alanines here, 1 alanine here, and 1 no alanine here. So total the amino acid occurrence for alanine is 4. Similarly, we calculate for all other amino acids, which gives the occurrence or number of these amino acids present in the given sequence. Coming to the composition, amino acid composition for a given amino acid, it will be like occurrence of an amino acid divided by length of the protein. If you see here, the length of the protein is 64 and there are 4 alanines. So when we want to get the composition of alanine in the given sequence, we do 4 divided by 64. It is 1 by 16 into 100. So we represent them in percentage. So the answer is 6.2. Similarly, if you take arginine, there are 3 arginines in the given sequence. So when we come to the composition of the arginine, so it is 3 divided by 64 into 100. Coming to this next question. So this is the given peptide sequences. So each amino acid has its corresponding weight given also. So what we do is multiply the molecular weight of L, molecular weight of E, molecular weight of A, D plus molecular weight of S plus molecular weight of E plus molecular weight of Q minus N minus 1 into molecular weight of H2O. Because these peptide sequence will have n minus 1 number of peptide bonds, which leads to the loss of water. So this is the formula how you get a molecular weight of a peptide sequence or a protein. So use this formula and try to calculate. I'll let you know the answer in a couple of minutes. there is no way to remember amino acid table. You have to know the short notations of amino acids. Even in the exam, most of the times short notations only will be asked. The concept of finding a molecular weight is nothing but we add individual amino acids weight and we subtract the water weight that is lost to the peptide bond. So if we have a 5 amino acid polypeptide, so there will be a 4 peptide bonds. That means 4 molecules of water is lost. That is 4 into 18. We have to subtract from the sum of the individual amino acids weight. So that is the concept here. However, more problems related to this we'll be solving next. I think these are the tasks related to the next week. So any doubts you can ask, otherwise you are free to leave. The session is done for today. Just give me a second. Give me a second, I'll turn on the mic for everyone. Yeah, I have allowed everyone. Whoever has the doubt, they can turn on their mic and speak. So the session for today is done. I thought I could take some more basics questions for the next week, since I'll be getting a next class only on next Monday. By that time, the assignment deadline will also be approaching. Yeah. I just wanted to just give me a second.